Welcome back to our series on genetics with our second video on Punnett squares. In the previous video, we talked about the different terms that we use in genetics. We talked about genes and alleles, dominant and recessive, homozygous, heterozygous, and genotype and phenotype. Now we are going to be figuring out how we can predict what children will look like, or also use what children look like and their genotypes to figure out what parents look like and their genotypes. And for that, we are using a tool called a Punnett square. This is used to predict offspring. So if you have a mother and a father, a male and female, you could predict one trait about that offspring. These are used for only one single trait at a time. You can use it for more, but that would be something slightly different than what I would be teaching you. We are going to be doing things called mono-hybrid crosses. Mono meaning one. So, we are doing mono-hybrid, which is one cross, one single trait. If you were to use it for two traits, then that would be called a dihybrid cross. And that would be two traits. So, monohybrid would be, take a mom and dad, you test for hair color. That is a single trait. You can predict hair color. If you want to test for hair color and eye color, that is two traits. That would be a dihybrid. If you want to do hair color and eye color, and whether you have free ears or connected ears, that would be three traits, and that would be a trihybrid cross. But we almost never do trihybrid crosses, even in college. For this, we are focusing mostly on monohybrid, with probably a video coming later on dihybrid crosses if we end up getting there. And that would be for two traits, which you would do the same way that you would do the monohybrid, but there would be a little bit more to keep track of, and it would be a little more complicated when we are reporting the results of what we found. So, for now, we are focusing only on the single traits, only on the monohybrid crosses. So, how to build a Punnett square? It is, as the name says, a square. Extremely surprising that the Punnett square is a square, I know. It then has a cross or plus sign in the middle of it to make it into four corners, four squares within the square. This is what we will use to either predict the offspring or use the offspring genotypes to find the parents' genotypes. How we would do that, I will show you. Let's use, from our last video, our hair example. We have a mom with brown hair. So, we're going to give her a capital B because it is dominant. Brown hair is dominant to blonde hair. We have a capital B. And we're going to say that she is homozygous for brown hair, which means she has the same allele, the same letter, both capitals. And we are going to say that dad has blonde hair, also homozygous, but it is homozygous recessive. Mom is homozygous dominant, dad is homozygous recessive. And since we are breeding them, we are crossing them, we put an X between them, showing this is a cross. So let's take our parents and bring them over here into the Punnett square. Now, it does not matter which parent you put on top of the square. Normally, the first one listed is the one that we put first. So, since mom is listed first, we will put mom on the top of the square. You take mom's genotype, which is capital B, and you take this first letter here, which is capital B, and you put it above the first square here, which is also capital B, because it is that first letter. Next, we take the second letter, which I will underline here, and move the second letter here. To show where we got it, I will circle the one that was circled from there, and I will underline the one that was underlined from there. So you know where each of those letters got moved to here. Next, we go to the father. We will circle the first letter and move it to the side here. And then dad's second letter. We will underline and move here. So we have capital B, and capital B, lowercase b. Are you able to see that? Let's check, make sure that you're able to see that on the video. You are able to see it, good. I knew I was getting close to the edge there, just wanted to make sure. Unfortunately, this one is kind of hidden, so we will move it down a little bit. It is still, however, in this position beside this square. So, how we are able to show what they do is everything above drops below. Everything to the side comes to the side. So this B drops in the two squares below it. And this B drops into the two squares below it. Next, these two B's we bring to the side, since they're on the side. This lowercase b is brought there, and this lowercase b is brought there. The two B's to the side. This lowercase b is then brought over just like the one above it. And that is all you need to do to make a Punnett square. So we now have the four possibilities, the four combinations that the parents' alleles can make. You have capital B, or capital B, lowercase b. Capital B from mom, lowercase b from dad. Another capital B from mom. Another lowercase b from dad. You will notice that each of these are the same. Now, we will report two things on here. We report the genotypic ratio and the phenotypic ratio. The genotypic ratio is what letters do we have. So for this, each of these squares is worth 25%. So There are 4 possible squares, 4 possible combinations. How many of them are capital B, lowercase b? All 4 of them. Which gives us a 100% capital B, lowercase b. Phenotype ratio, what they physically look like. 4 out of 4 have a brown and a blonde, and since the brown is dominant, that leaves us with 100% will have brown hair. And that's all you need to do with a Punnett square. Hopefully this helps clear up any confusion about how we get the letters and how we read it here. Our next video, we will be doing different types of Punnett squares. Simple Punnett squares like this. We will be doing just a video of practice on it. If you feel you already completely understand this and do not need any more practice on the Punnett squares, feel free to skip to video 4, in which we will discuss incomplete dominance and codominance. What happens if instead of 1 capital letter and 1 lowercase letter, if you have 2 capital letters? What does your genotype look like? What does your phenotype look like? That will be video 4. If you still need some practice, video 3 will be practice on different Punnett squares.